Good morning. I'm Mady Hornig, and I'm a researcher at Columbia University, working at the Center for Infection and Immunity at the Mailman School of Public Health. I direct translational research there and have a very strong focus on ME/CFS and trying to understand the mechanisms that cause this disease. Our interest has been to try to understand the causes of ME and to use every bit of knowledge that we can comprehensively to determine the causes of this disease. We don't think that it is a singular cause, so I have specifically used the word causes. And we think that just like a disease like cancer is a process whereby cells are dysregulated and multiply out of control, but it's not just one disease. There are many different organ systems that are affected with cancer, and there are many different processes that lead to the disease. We think that that's very similar in ME, that there are multiple causes, multiple pathways that may be important, and that we can use the clinical features in specific groups or subgroups of patients to help us understand these different pathways and these different triggers. Some triggers may work through a common pathway and may lead to something that looks very similar. So it may be that there may be a viral infection that may lead to very similar effects across patients, but it may not be the same virus for each of those individuals. So one common virus, for example, is Epstein-Barr virus. That's the cause of what I think you refer to as glandular fever here. We call it infectious mononucleosis. And about 10 to 15% of individuals who contract glandular fever will go on to a prolonged course with an ME-like scenario. But other types of infections may also lead to a similar type of finding. So HHV6, human herpes virus 6, also can lead to some similar types of scenarios. So in the first weeks of those infections, let's say HHV6 and Epstein-Barr virus, there may be some different characteristics. Some rashes may happen in HHV6, and there may be some other types of features in Epstein-Barr virus. A sore throat and swollen glands may be much more prominent. But over time, after the disease has become more chronic and turned into an ME-like picture, there may be much more similar patterns of symptoms. But we do know that this is a diverse disease, and it's probably the case that viruses are not the only contributor, and there may not be, again, it's unlikely to be a singular cause. One particular area that we're really looking into very closely now is to see whether there may be some features in the gastrointestinal system that could help us to understand at least some part of the disease, and could also perhaps give us some clues as to how to improve or even reverse the situation. About anywhere from 30 to 70 or 80% of patients with ME are thought to have symptoms associated with irritable bowel syndrome. So they can have constipation, diarrhea, gassiness, nausea, a whole host of symptoms that are very uncomfortable and that may relate to abnormal gut bacteria. Now, this is the microbiome that we talk about, you hear about in the popular press. People are discussing what probiotics they may take to improve the situation. Recently, we found that there are some links to a disturbed microbiome in ME that are very prevalent among ME patients who have a diagnosis of irritable bowel syndrome, as opposed to patients with ME who do not have a diagnosis of irritable bowel syndrome. So we're exploring that. We're trying to understand what these different bacteria that make up those microbiome differences may do. They probably put some chemicals into your bloodstream that then go on to affect your brain and your nervous system in different ways, and can affect a whole host of organ systems. They may also influence your own metabolism. So the products of these bacteria may cause your own metabolic machinery to be impaired. And so we're trying to define what those differences are, at least in that subset of patients with irritable bowel syndrome, this phenotype, and to see whether we can design some treatments, probiotics, or other types of manipulations that may be able to address that. We also know that there are many other features in ME/CFS that, again, don't affect 100% of patients, and that may come in different constellations. Many patients with ME also have allergies, both hay fever or other types of allergies that affect their comfort. They're maybe always having a runny nose, runny eyes. They may have itchy skin. There are a whole host of these types of phenomena. And so we're looking very carefully into that to see whether that can identify certain types of biomarkers. Now, do we think that allergies are a cause of ME? Maybe not. But individuals who are vulnerable to developing allergies, either because of their genes or their environment, perhaps they're in an environment with mold or pollens, so it may be seasonally worsened by what's in the air. We know that there may be some important factors, at least as a trigger or something that will tell us something about that particular individual's pathological processes that are leading to the disease. So, for example, there are cells that are involved in allergic processes. These are called mast cells. And these mast cells are full of a chemical called histamine. And so we have effects of those cells and other immune molecules that are induced by the changes in these, in these allergy-related cells and their products. And so we are trying to tie these things together within the subset of individuals who have allergies. So we're looking at their immune markers, we're looking at their metabolomic features, the metabolites in their blood system that may affect their nervous system functioning both in their brain and in their peripheral nervous system, causing pain, perhaps sensory pain, muscle pain, also gastrointestinal pain. So we are really, I think, making some good headways with this by having large number of patients, having some opportunity now to look at patients over time, the same patient over time, and marking their clinical problems, rating how severe the different problems are, from the brain fog to the allergy problems to the muscle pain to the energy levels and the overall physical fatigue, post-exertional malaise, all of these features rating over time, and then looking at how their blood markers and their microbiome are changing in concert with those differences. We know very little about what we call the natural history of ME. And so we're trying to define that. We've just completed a 1-year look, so we have 4 time points in 125 patients and 125 controls. And while we haven't looked at the blood samples or the bowel samples, the fecal samples yet, to understand, the biology, we can learn a lot from how the disease fluctuates over time. Are there seasonal changes? Maybe some people are very static and never changing, and other people may have much more of an oscillating course up and down, and again maybe seasonally relating to their degree of activity and so forth. So we want to understand that better, and we now have collected data and are really using that data to hone down, do what we call data mining, to try to understand how the different symptoms fit together and what it can tell us about the different subsets. How do we use these data to define different subsets in ME? Who has the equivalent of a blood cancer versus a lung cancer? Who has, with ME, who has the allergy type of ME, and who has a very prominent brain fog type of ME that perhaps has a different set of causes and a different set of treatments. So that's really, I think, it's precision medicine on a public health level. So it's trying to, we want to be individual, but I think we really need to identify the subgroups that are important for understanding causes, perhaps even ultimately preventing disease, but really making an impact on the disease process. So we're getting closer to that. It's actually, it's become extremely exciting, and we use many different sources of data, many different types of biological samples, and eventually we hope to include more in physiologic testing, neuropsychological testing, brain imaging testing, as well as other types of tests that can give us a better, more comprehensive view of these different subgroups, because we don't think that everybody is going to have, there's some interest in what's called small fiber neuropathy. So these are small, unmyelinated fibers. They don't have the typical coating, like the electrical coating that myelin usually provides. These are involved with pain syndromes that may be not only in the skin and in muscles, but also in the gastrointestinal tract, and may help us to understand the wide variety of symptoms that are present in ME, some of which may be common across the subgroups. These fibers can affect what's called the autonomic nervous system. We think of that as our fight or flight system. It's the sympathetic nervous system and the parasympathetic nervous system. And in an area that is very close to my own research, I'm tremendously interested in how the body, the immune system may become subverted to attack those small nerve fibers and other nerve targets, nervous system targets, by producing antibodies that latch on to the functional parts of these nerve fibers or other brain and nervous system targets and disrupt their function. That particular type of model is one that I find very encouraging if it really holds up, because it's very tractable. An antibody has what's called a half-life. It has a half-life for binding also, so it'll bind to its target, but it won't stay there forever, then your body breaks these antibodies down eventually. So if you can affect the new production of these problematic autoantibodies that are affecting the nervous system components, you have something that's tractable. You have something that you may be able to reverse and perhaps even to cure if those antibodies haven't caused damage or to the cells. That's part of the principle of behind the treatment with rituximab, which many of you have heard of. It's a monoclonal antibody that has been designed to help to get rid of a certain antibody producing cell called a B cell that makes these, that can also make these offending antibodies that are potentially associated with ME. We're also finding that the gut controls whether those B cells will make those bad antibodies or limit themselves to good antibodies that help you to fight infection and so forth. So again, tying that all back, even though there's so much diversity, there may be many roles that the gut plays, even in controlling this autoantibody problem that may be involved in a subset of ME. And it's one in which I'm focusing my attention because I think that it's highly tractable, maybe multiple ways besides something like rituximab, maybe other treatments that may also help us to either limit those antibodies that are causing problems or eradicate them altogether. And it's possible that we may be able to do that through the gut, potentially, because those antibody-producing cells are migrate through your gut every day. And so they are taking their signals from the bacteria that are in your gut. So this is really sort of one of the areas that we're looking at. Again, it's probably only going to explain a certain portion of ME, but we think it's a tractable one and it's very promising. So as we define these different subsets, we will be able to identify some of these different pathways that are going wrong and be thinking about how to address them therapeutically or even prevent further episodes after getting rid of the bad antibodies.
Be feasible?
Yeah, so fecal transplantation is clearly one treatment pathway that holds promise. My concern is that we may not know enough yet about what type of microbiota we should be putting in to an individual. That's where I think we are at a frontier. I think that my impression from where we use fecal transplantation in the United States now, it's only really used officially for a certain type of infection that is very severe called C. difficile, Clostridium difficile. It is an infection that becomes antibiotic resistant, and the intestines of individuals who have this infection become very destroyed and almost like lace, and that it's a terribly destructive infection. What is remarkable to me is that fecal transplantation is very well tolerated with very few adverse effects, not zero, but very few adverse effects, even when you're putting it into the intestines of people who have so much damage. And usually it's done in children also in the United States. It's one of the common subpopulations where we're using the fecal transplantation now. There are a few other studies that have been done in other circumstances on a research basis. What concerns me is that it doesn't stay. It doesn't usually remain for a very long period of time. With C. difficile, it does seem to prevent further infections. And it may be the difference between an infection where you have a huge overgrowth of a particularly bad bacterium, as opposed to what we call dysbiosis, which is an imbalanced bacterial grouping in your intestines. And so we need to understand more through the fecal transplantation, which, from case reports, you hear many positive reports of initial effect. I haven't heard that many reports of sort of curative effects. So I guess it's possible that one could potentially keep on doing fecal transplants, but who do you use as your donor, and how do you decide which? We know that the microbiome of healthy people doesn't always look exactly the same. So you can have, two groups of healthy individuals with very different microbiomes. They are not, just because they're different doesn't mean that they're abnormal. They're, both healthy. The question is, how does it work for them, right? So maybe their genetic situation or their diet is one where, they are eating a diet that is complementary to their microbiome, or their microbiome becomes complementary to their diet. We don't know enough yet in my mind. I think it's very important to pursue and to monitor and to understand not only how the microbiome changes over time after a fecal transplant, but perhaps also looking at the blood metabolites that are both from the bacteria in the gut, as well as from what the bacteria induce in the human, because there, you want to see whether there are positive changes there, so we can understand more about what's happening and continue having people rate their health on multiple, what we call domains of function, different organ systems, different types of problems, and to really understand that, and perhaps also adding in a little bit of physiology. We have so much now with mobile technology. We can monitor heart rate. We can, look at, position that people are in. We can look at, electrical conductance in the skin, what's called the galvanic skin response. Gives you a little bit of a sense of the sympathetic nervous system, which is your sort of your fight or flight system, and versus the parasympathetic system. So we have many opportunities now to integrate these. So I do think that fecal transplantation may ultimately be one way to go. I think also that there may be ways to use various forms of probiotics, maybe not even live bacterial agents, or prebiotics, which are the foods that feed the bacteria. So there's many different ways to go. One particular interest of mine is use of diet to change microbiome, and particularly to affect inflammatory or autoimmune problems. And there's some extreme diets that seem extreme, but they are used for very severe conditions like autoimmune epilepsy, where drugs don't help anymore and people are having, like 100 seizures in a day. They go on certain diets. One is called the ketogenic diet, and after a period of years, some of them become medication free and they no longer have seizures. And it's an autoimmune problem. And so we know that there can be very powerful effects from diet. We don't want to encourage people to go off and do something that might be unhealthy, but it's worthy of study when there are such really markedly prominent responses in people who have severe clinical disease. And again, a seizure is easy to see and monitor. ME, of course, has the huge difficulty of being somewhat invisible. And so the more we can monitor and make this disease visible and show how various interventions change those different physiologic and clinical symptom patterns, the more we're going to be able to get some real traction in this disease.
Subgroup to subgroup, what would work?
Absolutely. I think that that's really what we need to try to identify. And when we have the opportunity to build in, find what people are eating, have them record their diet in addition to testing their gut microbes and how they change with the diet and how their metabolism changes associated with those dietary and microbiome changes, we're going to really be able to build our knowledge here. I think that that's really one of the key phenomena. So there are both genetic factors. I mean, some patients can't tolerate gluten. From a genetic perspective, they have immune genes that are contributing to that type of response, and so there's sort of a precision aspect to that. We really do want to understand how these phenomena occur. We probably do need to build in some more genetic studies and also look at gene expression. We have some systems now that we're able to do to potentially look at those food responses, at least from the blood cells, and see how blood cells respond to different types of food components. And so sort of test tube situations, it's a system where you can pull the blood into a test tube that has some type of dietary component in it. And then you can look to see how the cells in the blood are responding to that. At the same time, you can also monitor changes in the microbiome. You can also look to see if there's evidence of a problem in the barrier that should keep contents from the gastrointestinal tract from going right into the bloodstream. Right, so there should be this barrier, and that what we call leaky gut is something that has an association with certain food-related allergies, and some of the allergies or hypersensitivities may not be from the food per se, but it may be what the food brings in with it. So there can be all sorts of things from the environment, including things that are getting into the food chain through pollution, so pesticides, organic pollutants, and so forth. These also have a very prominent effect on the immune system, can increase the production of antibodies that are problematic and interfere with the production of antibodies that are helpful and help you clear infections. So there's a lot to be learned there. There's throughout the food chain, throughout the world, even polar bears have flame retardants in their bloodstream. And so we know that it's all throughout the food chain. And so we are learning about the range of effects that can occur. And again, because your diet is, exposes very long system in the intestinal tract. It's a lot of real estate inside your body. And so there's a lot of opportunity for exposure, particularly in areas like the small intestine, one of the most highly metabolically active areas. And there are antibody-producing cells that are trafficking through there all the time. So it forms this intersection of your the environment with your immune and your gastrointestinal system and the bacteria that are in your gut. Some bacteria may help to detoxify. They may be helpful and may address some of those toxicants that are coming in through the diet, and some may cause more problem. So, we really need more knowledge, on this. I think that while fecal transplantation has shown itself in certain situations to be very safe, it also seems to be very wasteful to risk losing hope for people if you are using a fecal transplant, but you don't necessarily know what the right way to do it is, or it's not done in a controlled, setting. We want to be certain that we also don't close off certain avenues because we want to be sure we're doing the research in the right way. So and not just using people's home remedies, as I know people in the United States who are buying a blender and making their own transplants, all done at home, and there's no great control over it, and then they say, oh well, it didn't work, but then we may close off an avenue that really could be extremely important and perhaps even curative for some individuals. We could close off those avenues prematurely. So I think it's a balance between finding a way to ensure that the pace of research is fast enough, but that we don't say, well, I tried a fecal transplant at home and it didn't work. We want to be sure that we have do it right. So. One of the models that I use to put together what risk a particular individual has of getting ME or any other disease affecting the brain is what I call the 3 strikes model. What are the 3 strikes? One is genetics. The other is the environment, environmental factors, infections and toxins, and perhaps even psychosocial stressors or trauma. And the third is timing. So when an environmental factor is introduced into a person, their genetic inheritance is in a particular state depending on where their maturation is. So again, in utero, before birth, there's a certain unfolding of genetic programs and certain genes that are active and turned on and off and capable of being affected at different times. And then throughout your life course after birth, that changes. And so we know that there are 2 time points where ME has its highest prevalence age-wise, and one is around 11, 13, so sort of the adolescent age group, and then the other is closer to 30. So we have 2 time periods where there's this vulnerability. What's happening maturationally, and which gene programs, genetic programs are turned on and off at those times. There are, of course, puberty is active around that 13, 11 to 13 age group, and there may be differences in sex hormones that are beginning then that may alter the course of this disease. But we think that that we need to understand more about the genetic factors and what we call epigenetic factors, which is the imprint of the environment on the genetic inheritance and controls which genes are turned on and off, in addition to the maturational effects on the turning on and off of genes. And that, I think, really helps us to understand disease. And then there's some diseases that are present and evident early in life, some that are typically in middle life. So ME is one of those. Also depression, multiple sclerosis, and then others that are in late life like Parkinson and Alzheimer's. And so we think that these processes are really important to understand. We're born with all of our genes. If all of our genes were turned on at the same time, we wouldn't be alive. So this process of environment, actually some environmental factors are important to help us to turn on and off, turn genes on and off at the right time point. Some are driven just by what we call developmental programs, but this process is something that we can read to some extent through gene expression. So the products of the genes, the middle products of the genes, which is RNA, and then also proteins that are made off of that template of RNA. And then you also have epigenetics, which is the molecules that are actually regulating which genes are turned on and off at one time. And that process may be different in your brain than in your heart. So there are local effects as well. So we're always adapting to our environmental factors through this process of epigenetics and gene expression, regulation of gene expression. And so these three strikes, I think, are really important to understand disease and to have an impact to control disease. We found early on that individuals who are early in the course of disease as opposed to later in the course of disease, and we used a 3-year cutoff. We found that the immune system was more activated in that early phase of disease. In later phases, particularly 10, 15, we had some people who were 30 plus years from the onset of their illness. There appears to be more of an immune exhaustion. And both of those have important implications for treatment. And so we know that we have some factors that we can use to stimulate the immune system, and that may be more important later on in disease. So compounds like Ampligen and so forth that boost the immune molecules and help to increase some of these immune molecules, because you need them for brain function. You have receptors for these immune molecules in your brain, in your stress response circuitry in your brain. So if you don't have the immune molecules, that's also a problem. Have too much, as we saw early in disease, you may want to have something that dampens down that. And so there are things like anakinra and other types of immune suppressive types of compounds that may be really beneficial. And then, if there are specific treatments that specific causes that one thinks may be really important, like a virus, there's also, opportunities to use things like antivirals as well. But the gene environment timing model gives us some sense of where a person is in the course of their disease, and to try to identify what might work best to help eradicate all of the features of their disease. And perhaps, could we use the term cure? But that's really sort of where we want to head.